Deoxyribonucleic acid, or most commonly known as DNA, is the genetic material that codes for the functioning of every cell in an organism. Prior to the duplication of a cell and its division into new daughter cells through either mitosis or meiosis, organelles and biomolecules have to be copied to be distributed among the cells. DNA must be replicated as the process ensures that each cell can receive the correct number of chromosomes. There are several steps associated with DNA replication, involving multiple proteins called RNA and replication enzymes. DNA replication works in the S phase of interphase during the cell cycle in eukaryotic cells and is crucial for organisms' repair, reproduction, and cell growth. Structure. To understand the process of DNA replication, it is important to understand the structure of DNA itself. DNA is a nucleic acid composed of a 5-carbon deoxyribose sugar, a nitrogenous base, and a phosphate. DNA, in double-stranded form, consists of 2 spiral nucleic acid chains that proceed to be twisted into the famous double helix shape, enabling for more compact DNA. Furthermore, in order for DNA to fit correctly within the nucleus, it is fitted within coiled structures named chromatin. Chromatin is a tight structure that is able to form chromosomes during the process of cell division. Chromatin also untightens itself so that cell replication can occur with availability to the DNA strands before the start of DNA replication. The setup, replication fork formation. Before DNA can start to replicate, the double helix must be opened and unzipped from a double-stranded form into 2 single strands. The opening of the double helix is carried out by an enzyme known as helicase, which is able to break down hydrogen bonds, allowing the holding and pairing of the complementary bases of DNA together, which are adenine, thymine, cytosine, and guanine. A is pairing with T and C is with G. The hydrogen bonding between the bases in their pairs is disrupted by helicase, which then allows for separation of the base pairs into the replication fork formation, shaped like the letter Y. This then becomes the foundation, or template, upon DNA replication to commence. DNA is then directional in both of the separate strands, that are signified by a 5 and 3 end. The numbering of the ends indicates which side group is attached to the DNA backbone. The 5 end has a phosphate group that is attached to it, while the 3 end has a hydroxyl group. The replication fork is bidirectional, meaning the 2 strands oppose each other in terms of direction. One of the strands is oriented on the 3 to 5 direction, which is known as the leading strand, while the other is oriented in the 5 to 3 direction, known as the lagging strand. Replication begins. Primer binding. After the separation of both DNA strands, primer, a piece of RNA, binds to the 3 end of the lagging strands as it's the easiest to replicate. Primers are generated by an enzyme named primase. The binding of the primer is the starting point for the process of DNA replication. Elongation. After primer binding, a new strand is created by a process called elongation, which enzymes known as polymerases are responsible for. DNA polymerases have 5 types in bacteria and human cells. For example, E. coli, a bacteria, has polymerase 3 as its main replication enzyme, and polymerase 1, 2, 4, and 5 have responsibility for repair and error checking. Polymerase 3 binds to the strand that is at the site of the primer, then starts to add new base pairs that are complementary to the strand during replication. Polymerases alpha, delta, and epsilon are the primary polymerases involved in DNA replication, specifically in eukaryotic cells. As replication proceeds in the 5 to 3 direction on the leading strand, the newly formed strand is then continuous as a result of elongation. Termination. Proceedingly, when the continuous and discontinuous strands are formed, RNA primers are removed from the original strands by an enzyme called exonuclease. The primers are then replaced with the correct bases. Another exonuclease reads the new DNA and checks for errors that need to be corrected. Moreover, a new enzyme called ligase joins chunks of DNA called Okazaki fragments together to form a single and confirmed strand. However, the ends of the linear DNA pose a problem, as polymerase is only able to add nucleotides in the 5 to 3 direction. The ends of the parent strands are composed of repeated DNA sequences that are named telomerase. They act as protective shields at the end of chromosomes in order to prevent other chromosomes nearby from fusing. The type of polymerase enzyme, telomerase, then catalyzes the synthesis of telomere sequences at the ends of DNA. After completion, the parent strand and its complementary DNA strand coil into the famous double helix shape. At the end of it, DNA replication produces two new DNA molecules, each with one strand from the parent molecule and one completely new strand. The ultimate result of DNA replication is two DNA molecules consisting of a new and old chain of nucleotides, hence why DNA replication is described as semi-conservative. Half of the chain is part of the new DNA molecule and the other one is just part of the old. DNA replication as a process has shown its crucialness to the modern world, reaching the extent of becoming prominent in modern technology. An example of this is DNA sequencing, which includes determining the four bases of the DNA molecule, which tells scientists the kind of genetic information that is carried in a particular DNA segment. Thus, DNA replication is an essential process that enables organisms to function, as its multiple steps prove that accurately.